Very good morning. You must have watched previous two episodes in which we have discussed introduction to microscope and introduction to section cutting. Now in this series, this is a third video and we are going to discuss today staining and mounting of a section. When you have a basic knowledge about microscope, then next step is definitely you must have some basic knowledge regarding structure, anatomy, which are different cells which are in the tissue, which are different tissues, type of tissue. After that you should learn how to take section, that is the art of section cutting. Previous video we have already prepared on this subject. Now subsequent skill which you have to learn is nothing but staining. Now once you have different sections in your hand and those sections you are going to collect with a brush, and these sections you are going to transfer in the watch glass, and in this watch glass you can add different stain can be anything. Generally first we use fluoroglycinol HCl 1:1 proportion, which is generally used for the staining of the lignified tissues, xylem and other sclerenchyma. All dead tissues will be stained by using fluoroglycinol HCl. Now after this staining, maybe here 2 to 3 minutes you have to wait, so that there will be interaction. Stain is basically a compound which is going to have a chemical or physical reaction with that content of that plant part, and it is going to highlight particular structure, so the difference will become more visible under microscopy. So after 2 to 3 minutes of the staining, you can have a wash with water to drain excess stain. Most of the time ruthenium red or any other stains, they are too bright, and you need to give wash with water, and finally you are going to add cover slip on this particular slide. See, this is the ideal location where you are going to put your transverse section, and this is cover slip. Before adding cover slip, you make sure you have washed excess stain. There should not be air bubble which are going to be present under the under the cover slip. So basically you can take a help of a brush or forceps, add one part of that cover slip, and generally slowly you are going to slide that cover slip on the surface preparation. So this is going to prevent bubbling, and do not press that cover slip firmly. You have to remove excess water or excess stain which are present here and there on the slide with help of tissue paper, and after that your slide is ready to observe under microscope. Now when this slide is ready under microscope, first thing, you should not touch lens and mirror of microscope with your hand, because our hands always contain some oily or greasy material which is going to hamper your field view. Then if you are putting this slide on the stage, I already told, make sure there are no stains, like we are using hydrochloric acid, and that is going to corrode most of the stage of the microscope. Now first thing, you observe from the side. Okay, minimum distance should be 1 inch, or after that 1 inch you start coming upward from that point. So one particular point, by using large or coarse adjustment screw, you are going to observe specimen, and after that keep adjusting with small or minor adjustment screw, so that you can have clear picture of your transverse section. Now see, first thing, you are going to observe your section firstly under 10x. So 10x is going to give you broader picture or complete, you can say complete vision. Suppose this is a stem, and this is the middle part, pith, and this is the bark or the outside part. So this schematic diagram you can draw with help of 10x. But if you want to go inside or you want detail, in that case you can have 40x lens, more resolution, and finally 100x or oil immersion can be used with help of putting the oil here. But make sure whenever you are going for higher resolution, you should not touch that specimen with the with your lens. So cover slip is there, then this interaction is going to be avoided. And then next thing you are going to do here, it draws schematic or temporary diagram of your TS, what you are going to observe under microscope. And once this diagram is there, this is the final diagram which you are going to draw in detail. And that diagram is very important. You are going to show different types of tissues and cells right from the periphery to the middle part of the transverse section. So these are a few things which you are supposed to do in staining and mounting of the section. If you do it properly, you follow the protocol, this is routine practical in pharmacognosy, and you are not going to endanger any problem, you are not going to suffer with any problem if you are doing it systematically. Watch glass, you are not supposed to touch with your hand, use brush, proper staining, wash that with staining, remove excess water and stain with help of tissue, add a cover slip and observe in this pattern 10x, 40x, 100x. First draw the schematic or the outer line which are the different tissues are there, and after that you can use different stain. Fluoroglycerol HCl is used, then you can use iodine for starch, ruthenium red, picric acid, Sudan red. So many different stains are there which can be used properly for different tissues. So I think this is the basic regarding staining and mounting and how to place your section under microscope. In coming videos we are going to discuss in detail for example TS of leaf, TS of bark, TS of root and all that. Thank you very much.